So good morning, everyone. Thanks for this opportunity. This will be a short presentation today just to update you, as Vincent said, on the progress our team has made since my last talk in June. So I want to first emphasize that this has been a team effort by the names listed here. Can you all see my cursor?
Yes.
Yes.
Okay, great. So just to sort of review why we began this project was because in our group we are a mix of domain scientists and data scientists, and we want to sort of bring community annotation to the cardiovascular community. So this great portal, Gene Wiki, developed by Andrew's lab, seemed like a logical place to start. And in order to appeal to domain scientists in the cardiovascular realm, we wanted to first build a sort of strong representation of high quality interconnected Gene Wiki pages. Can you see that? Sorry, my clicker is weird. See the first? Okay. Of high quality interconnected Gene Wiki pages for genes expressed in cardiac muscle, with the idea that this will elevate this resource in the eye of domain scientists and attract them to this resource. So the way that we do this is to break the cardiac cell down into functional subproteomes. So these are manageable protein and gene families within which meaningful relationships can be annotated. So we have expertise in our group in cardiac, mitochondrial, and sarcomeric biology. So what we see as the great value of this approach is that it enables rigorous validation of the scientific content to ensure knowledge of high accuracy, depth, breadth, usefulness, and reusability. Excuse me. Excuse me. So since I have a little extra time today, I figured I would briefly describe some cardiac ultrastructure for those of you that may not be used to viewing this type of image. So this is an electron micrograph of a longitudinal section of a cardiac cell. So cardiac muscles are relatively large. They're terminally differentiated cells. They're about 20 microns in diameter, about 100 microns in length. Right away you can appreciate our two subproteomes of interest. So these are mitochondria. They're sort of sandwiched between these sarcomeres. So this is a sarcomere here. It's the functional contractile unit of a cardiac cell. So these are, these two proteomes comprise the most abundant subproteomes by mass within the cardiac cell. So within the mitochondrial proteome, we targeted cardiac expressed genes for 5 different functional categories. So there's metabolism, apoptosis, redox, proteolysis, and oxidative phosphorylation. So then sarcomeric proteins, these are the contractile machinery of a muscle cell. So for purposes of the Gene Wiki gene list, I'm including those proteins involved in longitudinal and transverse mechanotransduction and signaling as well. So these would be proteins comprising the Z disk, which are the dark bands here that sarcomeres attach to. I'm also including proteins comprising costameric structures, which these couple the Z disk to the sarcolemma here. The sarcolemma is the extracellular membrane of a muscle cell. So also proteins comprising intercalated discs, which is here, these sort of are, these link adjacent myocytes to one another in the actually their function is to electrochemically couple adjacent myocytes, I should say. So here's the breakdown of how I functionally classified the proteins and the number of proteins in each. So, okay. Basically, our protocol for improving these articles is to, this is review. I presented this before, so I'll breeze through this. So we create pages in BioGPS for those lacking subarticles, and the database content is automatically presented in the protein box using the PBB template developed by Andrew's lab. So next we do intensive literature searches and populate a list of journal articles relevant to gene and proteins. Then we organize this information into these sections here: introduction, structure, function, clinical significance, and protein-protein interactions. Then we write each section, And then we wiki link diseases and concepts, for example, between Wikipedia articles. And then we put a significant amount of effort into extensively citing references from PubMed to facilitate knowledge access. So to summarize our progress since my last presentation, we have more than doubled the number of improved sections, pages completed, references added, and wiki links added. Blue is our progress as of June, and red is our progress as of December. So you see this has been a substantial effort this year for us. So in bullet point format, we've improved nearly 1,600 sections across 387 pages. We've added over 3,700 references and over 10,700 links, totaling just over 2,600 kilobytes of content. So this puts us at a 62% completion rate for all the cardiac genes expressed within these two subproteomes, basically. So this kind of concludes this portion of the talk. I'm going to switch gears a little bit. I'm going to hone in on the Sarcomeric subproteome, which is where my sort of expertise lies. So I'll start by saying that a great motivation for building high-quality knowledge pages on sarcomeric genes was due to the fact that mutations in sarcomeric genes are so strongly linked to cardiomyopathy. So shown here is a Venn diagram by Jessica Lee. She's an undergrad student in our lab. Actually, I should say a very highly talented student in our lab. I have no idea how she drew this, but she managed to. So this shows overlap of sarcomeric proteins that house mutations in various familial cardiomyopathies. So what's not appreciated here is that roughly 50% of all cases of hypertrophic cardiomyopathy have been linked to mutations in sarcomeric proteins, and about 25% of dilated cardiomyopathy cases have been linked to sarcomeric mutations. So in fact, hypertrophic cardiomyopathy is now referred to as a disease of the sarcomere. So this work has high clinical relevance. So one feature that sort of sets this group of proteins apart from most is that they are mechanical proteins, and the insolubility of the contractile apparatus at a physiologic pH and its overall sort of structural resilience lends itself to being easily isolated. Experimentally, you can apply harsh detergents to solubilize membranes and all the soluble factors within the cell, leaving just the sarcomeres, which you can then bathe in sort of exogenous solutions containing different amounts of calcium to activate force and measure function. So shown here, this is a human cardiac cell. It's been permeabilized using detergent. When you mount the cell between a force transducer and a motor arm, you can set the sarcomere length to a normal resting value, and you can measure the isometric force produced at varying amounts of calcium. So this is a typical sigmoidal relationship that you would observe when doing this. And from this, you can glean certain functional parameters, such as myofilament calcium sensitivity, maximal tension, and cooperativity of activation, which is the slope of this relationship here. So why is this important with respect to sarcomere mutations? So these measurements, it's important because these measurements provide a straightforward assay of functional effects of sarcomeric mutations, and these are thus an important body of data to capture from the literature. So with the ultimate goal here being able to develop strategies for automating this process of building integrated knowledge pages from unstructured text data in PubMed, so once I finished all my sarcomeric gene wiki pages, I began to categorize all of the different biomedical entities or features represented on each of the Sarcomere Gene Wiki pages in an effort to identify these textual features that enhance the effectiveness of sarcomere protein information retrieval for different biomedical questions. So with the end goal of building and optimizing a dedicated, I guess you'd call it, cardiac sarcomere terminology for retrieving informative abstracts. So at least from my perspective as a domain scientist, and I'd love to hear all of your thoughts on this, the ultimate question to tackle here is, how do we extract information from text while preserving its wonderful complexity and richness? So my initial answer, at least to this, is mine is to mine high-quality phrases that provide specific and nuanced information. So what properties make phrases high quality? So listed here are some features that I thought about when doing this analysis. For example, popularity. So this is sort of respecting the common jargon in the field, right? So how are concepts commonly worded? So cross-bridge cycling kinetics is a very common phrase you'll see in all these, in this body of literature, versus something like kinetic properties of the sarcomere. So then another feature would be informativeness. So there are distinct types of tension generated by the heart. So simply saying tension is insufficiently informative. Instead, saying active tension or passive tension would be better. So then there's completeness. So saying calcium sensitivity, that's incomplete. Calcium sensitivity could refer to mitochondria or other organelles. Myofilament calcium sensitivity, on the other hand, will target the text we desire. So this is just a very preliminary analysis that I've done. It's a simple ranking of functional concepts specific to sarcomere proteins that would be relevant for extracting information on sarcomeric cardiomyopathies. So this just shows the popularity of these functional concepts among cardiac Gene Wiki pages. This is, of course, not exactly representative of the Medline database, but I see it as sort of a test set or sort of a reasonable starting point to begin identifying these phrases. So cross-bridge cycling and actomyosin ATPase activity, for example, were representative or present in 40% of these pages, and so on and so forth with the rest of these. So this basically concludes my talk. I would love to sort of brainstorm as a group. I'd also appreciate any questions and comments that you may have. And these are all the people to thank for all of this work. And yeah, I'm happy to take questions.
Yeah, this is Andrew, so maybe I'll start. Can you just go into a little bit more detail what you imagine the utility of sort of this this phrase, this informative phrase analysis in the Gene Wiki page, and whether you think, sort of off the top of your head, whether that aligns with what's in the literature?
Well, your first question was what I envision the utility of this.
Right.
So eventually, so this is sort of my initial manual analysis. I feel like after writing these Gene Wiki pages, I've gained a very good, it's a silly word, but I can't think of a better one, understanding or shall I say, because I've looked at, thousands and thousands of references of this topic, I feel like I've gained sort of a good understanding of the jargon used and sort of the wiggle room, if you know what I mean, within those phrases. And so in writing these Gene Wiki pages, I feel like that's reflected within those pages. Does that make sense?
I think so. So you mean that these are essentially the key functional terms that characterize the functions of these genes in sort of the cardiac setting? I guess what I'm driving at is that, how do we make this beyond sort of a standalone analysis? Would this lead to enhancements in the gene ontology, for example, in terms of how we categorize these terms from increasing generality to increasing specificity? Does it make sense to try to incorporate that into something like the gene ontology?
Okay, so I think, yes, I think it does. As I look through the gene ontology terminology, they're, understandably so, they're less specific than, some of these functional terms that I've shown you today. And I feel like the nuances with regard to, the effects of these sarcomere mutations are found within these complexities, these functional complexities. And so I feel like this specific information is valuable in that sense. Is that what you're asking?
Great. Sorry. So maybe it's a discussion we can continue offline, actually. But I think, one thing, because you guys have done such great work as domain experts, actually, even if gene ontology is at a more general level, right? It's because they don't have that domain expertise. So I think the Gene Ontology Initiative would welcome sort of the deeper domain expertise if we could translate the work and these general impressions you've gotten from looking at so much of the literature at this point into things that could be translated to the gene ontology. Then that could be made available through gene ontology annotations, and then other people could, other data scientists, right, could take advantage of this structure that that's emerging from your effort.
Yeah, thanks for that. I agree totally. The other thing, actually, since I have you on the line, is I noticed in the information box on each of these pages, there's Go terms, but there aren't any disease ontology or phenotype ontology terms. Is this something that we can somehow integrate into these pages? I think that's very useful information and could easily be.
Yes, and this goes to the other half of the Gene Wiki project, which I mentioned before, which is the use of wiki data, right? Okay. And this goes back to, how you introduced it really well. I think this interplay between domain experts and data scientists. And wiki data is really sort of our migration towards creating something that the data scientists would use, because of course text mining out of free text in the Gene Wiki is actually pretty difficult. So in the same way, I guess I'm suggesting that we could translate your work from free text into gene ontology terms. The other thing is to propagate everything that we do into wiki data. And the advantage of doing that is that, as you point out, then the Wikipedia, for example, if we added disease ontology and phenotype ontology terms to the wiki data entries for these genes, then we can pull them into Wikipedia, which is great. But not only that, it opens it up to querying by the rest of the data science community, right? It's sort of this massive community database. So that project is actually really, or that sub-project, I should say, has really taken off in the last, I don't know, 3-6 months. And, I'm really bullish on that. So what it needs, right, is like a lot of these data science efforts, right, it needs the motivating use case, right? What is the grand integrative query that you would like to do as a domain expert, but can't currently do? That combines gene ontology terms, combines phenotype terms, combines maybe epidemiological data, combines molecular data. So getting all that data into Wikidata and then coming up with the questions that might lead to novel insights, those are the things that I think we could work on. Yeah. Okay. Awesome. Thanks.